Good morning to all of you. How are you all? How are you all? Are you able to listen to my voice? Okay, so my voice is audible to all of you. Yes, good morning, Anjali, Archana, Merlin, Radhika. Please let me know my voice is clearly audible to all of you. Okay, okay. So yes, welcome for today's session. Okay, okay. So I am welcoming all of you. I take the today's session. Uh, what is the timing? Basically, yesterday, which timings I provide you yesterday? I think I told you that we will take the session around 11 p.m. Sorry, 11 a.m. So I already told you this in the last lecture or not? Please let me know. Because sometimes I forget to, I have so many students, so many batches. So I definitely forget. But please let me know. Okay, okay. So I already give you the timing yesterday. Okay, okay, okay. Then it's okay. Because sometimes I forget the timing which I provide to this session. So we have our lecture from 11 to 1 p.m. Okay, okay. So thank you a lot, Radhika. So we have to take your session from 11 to 1. Okay. Okay, just waiting for 1 or 2 minutes more so that all the other students will join. Otherwise, we will start our session and they will go through the recorded version or they will go through the link. Then they will be free because I know today is Sunday. So we all will have resting time today. So that is why if they will not be able to get it right now, they will be able to watch it later also. Okay. I already give you some homework also. Only Anamika and one more student is here who is able to answer. Yeah, the functioning of serotonin, dopamine, melatonin, and histamine. So I am waiting the answer from all of you, but only Anjali and one more student. I don't know the name. What's your good name? Maybe Anjali. Yes. So Anjali and Anamika is the only one who just completed the homework, otherwise all of you are not able to answer. So why you are not able to answer the question which I gave you yesterday? Please let me know. So please let me know the functioning of serotonin, histamine, dopamine, melatonin. So I already give you this homework, but only two students are there who answer the question. So this will not be tolerated. I will ask the question every time. So you have to do the homework, okay? Otherwise, how I will be able to know that whether you are getting my points or you are not getting it? So it's my request to all of you that please complete your homework as soon as possible. So yes, Asutosh, good morning. How are you? So yesterday we have done this protein hormones, right? So we have completed the protein hormones. Today we will focus on the steroid hormones. Okay, so I will start with steroid hormones because we have completed the protein hormones yesterday, and also I have to start thyroid hormones because we have to discuss disturbances in the thyroid. So for knowing those disturbances, what we must know what basically is the thyroid hormone, what is the functioning of that particular hormone or this particular gland in our body. So we have to know about the thyroid in detail. Then we will able to know the disturbances which are there. Getting my point? So please complete your homework. When I give you some homework, then please take it seriously. Okay. So I also provide you lots of work today also, and I appreciate if all of you will be able to answer me. Getting my point? So I am just going to start the session. So please let me know my voice is audible to all of you. Only once please let me know. Otherwise I will able to start the PPT now. So my voice is audible to all of you. So yesterday we have done this protein hormone. Yeah, we have completed this, no? And I already provide you the PPT of this. So today we will start the steroid hormones. So my voice is audible to all of you. Okay, no issues at all. Take your time. No issues, Ayushi, when you will be able to share the PPT, you will be able to share the PDF basically, you can, but please try to do the things, okay, so that you all will be remain in touch with the topic. This is only the concern which we have. Okay, so I am audible. So I am not, now I am just going to start steroid hormones, and we have to complete it today. Okay, thank you all. Thank you, Pooja. Thank you all. Okay. So today we have to start steroid hormones. Also we have discussed some important points which are there in the steroid hormones during our previous lectures, but we have to discuss it in detail. Now, what are basically the steroid hormones? What is the functioning of these hormones? Where they are applicable? How they are able to perform the particular type of work, or particular type of functioning basically. So, steroid hormones are the crucial substances for the proper functioning of the body. So, for the proper functioning of the body, we need the steroid hormones. So, steroid hormones are crucial substances for the proper functioning of the body. Steroid hormones, they are produced in the adrenal cortex, testes, ovary, and some peripheral tissues. So, from the adrenal cortex, testes, ovary, they all are the areas from where we are able to get the steroid hormones. This is the point. All steroid hormones are derived from cholesterol. We have discussed it in previous lecture also, that steroid hormones are the derivative of cholesterol and differ only in the ring structure and side chains attached to it. So these steroid hormones, they differ only in the ring structure. So they are the derivatives of the cholesterol, number one. You have to keep it in mind, and differ only in the ring structure and side chains attached to it. So these are the steroid hormones. So enzymes which produce steroid hormones from cholesterol are located in the mitochondria and in the smooth endoplasmic reticulum. So steroid hormones definitely produce some important enzymes or some important from cholesterol, which are located in the mitochondria and in the smooth endoplasmic reticulum. Okay, the main point is this, that steroid hormones are derived from cholesterol. They are the derivatives of cholesterol. Another important point is this. All steroid hormones are lipid soluble and water insoluble. We have already discussed it also, that the hormones are basically steroid hormones. They are lipid soluble. So that is why they are permeable to membrane, so that they are not stored in cells. This is very important point. Like protein hormones, the protein hormones are pre-prepared or they are stored in the vesicles, but they are not stored in the vesicles, rather they are produced or released immediately during need. So it is permeable to membrane. These steroid hormones, they are lipid soluble, so they will be allowed to pass from the membrane and they are not stored in the cells. So these two are the very important point of steroid hormones. They are derived from cholesterol. They are the derivatives of cholesterol. Number two, they are lipid soluble. They are water insoluble. They are not stored in vesicles. Rather, they are produced immediately. So, steroid hormones produced in the adrenal cortex, testis, ovary. So you have to keep this point in mind also, that steroid hormones are generally produced in the adrenal cortex, testis and ovary. So all steroid hormones share a typical ring structure because they are the derivative of cholesterol. So yes. All steroid hormones are derived from cholesterol and differ only in the ring structure and side chains attached to it. So they are basically cholesterol, but they have some little bit difference in their side chains or ring structure. Otherwise, they are similar as we have the cholesterol. All steroid hormones are lipid soluble. Now next point is, steroid hormones are non-polar and can thus diffuse across lipid membranes. So these steroid hormones are non-polar and they can pass from the outer membranes and they can directly enter into the nucleus also. So if this is a cell, this is a nucleus, so they will be able to pass from this outer membrane and these will come here up to cytoplasm and even in the nucleus. So they leave cells shortly after the synthesis. Now these are the types of steroid hormones which we have to study. We have glycocorticoids, mineralocorticoids and androgens. So let me see whether you are getting. So everything is clear up to now. Yes, good morning to all of you and no issues. Just be there along with me. So are you getting it? Okay, and I request any one of you to please share the link once again in the group so that the another students will also able to join. So I request any one of you, please share the link again so that they will join also. As we have already discussed it in yesterday that we will start the session from 11 a.m. But they are not still here. I don't know why. Yes, I remind that I wrote 11 p.m. but it's a.m. So I hope all the students should be here. Otherwise they will watch the recorded video. Okay. So now I am just moving further. So we have to discuss the types of steroid hormones, the important types of steroid hormones. We have glycocorticoids, we have mineralocorticoids and we have androgens. Now what is basically glycocorticoids? These steroid hormones, they are originated in the adrenal cortex. Like we have already discussed here that steroid hormones are produced in the adrenal cortex. So These are those hormones, these are the glycocorticoid hormones which are originated in the adrenal cortex and affect mainly metabolism. So generally they are produced in the adrenal cortex and affect mainly metabolism by decreasing inflammation and increasing resistance to stress. This is the very important point of these glycocorticoids, that they increase resistance to stress. And the very common example is cortisol. So in the very first day I asked you to write the functions of cortisol. So this is the main function of cortisol, the hormone which is produced by the steroids, or you can say by glycocorticoids. So this cortisol or this corticosterone or dihydrocorticosterone or cortisone, all these are the examples of glycocorticoids which helps us to to bear the stress. Getting my point? So cortisol and cortisone, or you can say all these glycocorticoids, they are helpful for us to bear the stress, to resist, to increase the resistance towards the stress. Because if you all are aware, you all are quite young now, you all know that everyone is speaking, I have so much of stress, I have so much of stress. Even you, when you talk with a child of 10 years who is just in first or second class, he or she also has some type of stress. Okay, so these stress is there only on the recent years. Otherwise, in the past years, the stress word is not that common as it is common right now. So everyone has stress. But these actually these hormones are the basic ones which are, you can say, the imbalance of these hormones are the basic one. That is why we are feeling these type of stress, sadness or depression. Okay, no one is want to do it, these things. Like if you are sad, it is from your inner side. If you are feeling depressed, it is from your inner side. It is from your inner heart. It is because of your inner hormones. Are you able to get my point? I want to say that if you have some imbalance in your hormones, if you have any type of imbalance in your hormones, it will definitely affects your whole health, your mental health, your physical health. So hormones play a very important role in our body. Getting my point? So we must have a good level of cortisol, we must have a good level of cortisone, corticosterone, dihydrocorticosterone, basically the glycocorticoids. So we must have a good level of these glycocorticoids to bear all those things which are there right now. Okay, so we must have the proper balance of hormones in our body. If there is any type of imbalance of hormones, either the level of hormone is more or the level of hormone is less, so we must have the proper balance of these hormones. Okay, so this is the glycocorticoids which originate in the adrenal cortex and they affect the metabolism. Hormones are the only one which have long run effect on our body. So hormones are the only one which have long run effect. Getting my point? So you must, so you don't feel, as I always told my students, to don't feel stress or anything like that. Always try to remain happy in every situation. Always try to keep your hormones in balance. Okay, so this is basically glycocorticoids. Now number 2 is mineralocorticoids. They are also originate in adrenal cortex and they maintain the water and salt balance in our body. So the mineralocorticoids, they maintain the water and salt balance in our body. We will discuss these in detail also. The very common example is aldosterone. Then another is androgens. Now what is androgens now? So androgens originate in the adrenal cortex and gonads and primarily affect maturation and function of the secondary sex organs, like testosterone is a hormone. So they are responsible for the growth of for the growth of sex organs in case of male as well as in case of female. So we have these hormones. These are the steroid hormones which are quite important in our body. The imbalance in any of these will leads to put a great effect on our body, even from inside as well as from outside also. Getting my point? Now again, glycocorticoid, mineralocorticoid, and we have the androgens. So glycocorticoids, in glycocorticoids we have cortisol, which is the main uh, hormone or the major representative in most mammals. Cortisol is quite important in our body because it is the one who regulates the stress level in our body. The another is mineralocorticoid. Mineralocorticoid, the example is aldosterone, and we have to discuss this for relating salt and water balance. So we will discuss it in detail, aldosterone, and in case of androgens we will study testosterone. And some another hormones are here which are estrogens, which are there in the female for controlling the during the female growth. These hormones are quite important. We have estrogen, we have progesterone. You must heard about all these. So we will discuss them in detail. So what are basically estrogens? They are also like we have androgens in case of male. So androgens such as testosterone, they are quite helpful in the growth of secondary sex organs in case of male, and in case of female we have estrogens. So in case of females we have estrogens which are originated in the adrenal cortex and gonads and primarily affect maturation and function of secondary sex organs. The example is estradiol and estrone. So all these hormones they are also originated from the cortex, and they are the main one which are responsible for the maturation and functioning of the female secondary sex organs. Then we have progestogens. Progestogen, or also known as progestogens, they originate from both ovaries and placenta, and they mediate menstrual cycle and maintain the pregnancy. So progesterone hormone is quite helpful during the pregnancy. The level of progesterone is high during the pregnancy. So are you able to understand up to now the things which I want to clear? We have steroid hormones. Some properties of steroid hormones are there, and steroid hormones are of three types: glycocorticoids, mineralocorticoids, and androgens. In case of females, it is known as estrogens. So glycocorticoids, which basically includes cortisol, which is resistance to stress. In mineralocorticoids, we will study aldosterone, and in case of androgens, we will study testosterone. So glycocorticoids, mineralocorticoids, androgens, and estrogens. Androgens in case of male and estrogens in case of female. So again, the properties of steroid hormones, that they are not packed but synthesized and immediately released. So they are not packed as they are packed in case of protein hormones which are stored in the vesicles. Rather, they are synthesized and released immediately. So they all are derived from the same parent compound, which is cholesterol. So enzymes which produce steroid hormones from the cholesterol are located in the mitochondria and smooth endoplasmic reticulum. These steroids, they are lipid soluble. That means they are freely permeable to pass through the membranes. So they are not stored in the cells. Okay. Another property is steroid hormones are not water soluble, so have to be carried in the blood complex to specific binding globulins. Yes, this is very important point of steroid hormones. As they are water insoluble, so what we have to do? They have to be carried in the blood complex to specific binding globulins. So they have to be passed through, or they have to be carried through blood, and in the blood also they will have to bind with some specific globulin molecules. Okay, so corticosteroids binding globulin carries the cortisol, so they have to bind with a globulin molecule in the blood also for passing. Similarly, sex steroid binding globulin carries testosterone and estradiol. So in some cases, a steroid is secreted by one cell and is converted to the active steroid by the target cell. Now it is the example, or you can say some modification of steroid hormones. The steroid hormone is secreted by the one cell and it is converted into active form by the target cell. So they are in the pre-active form, but later it gets converted into the active form. So an example is androgen, which is secreted by the gonad and converted into estrogen in the brain. So they are secreted somewhere else, but they become active on their target cell. Getting my point? So sometimes what happens, these cells, they are just released as inactive form, or you can say they are just released as proactive or inactive form, and they will become active only when they will reach the target cell. Okay, so they become active only when they reach the target cell. So this is also another property of steroid hormones. Now this is the structure of some corticosteroids like cortisol and corticosterone. You have not to remember these structures. You have to keep it in mind that they are derived from the cholesterol. So they have little bit modification in their ring structure only. So if you are able to see here, we have everything is common. 11, everything is common. The difference is here only. So here we have C double bond O, CH2OH. Here instead of C double bond O, we also have OH group. So only a little bit difference in the ring structure occurs in the steroid hormones. This is again 11 dihydrocorticosterone. Only little bit modification is there only. Okay. This is corticosone. Only OH group is here. Now this is aldosterone, which is basically the mineralocorticoid. This is testosterone. So if you will be able to see here, only few, like here we have C double bond O, CH2OH, CH double bond O. So only a little bit difference in the ring structure only. So this is estradiol. This is again progesterone. So up to now, I hope you will be able to understand about the steroid hormones, the functioning of steroid hormones, the hormones which are there in the steroid hormones, what are the functions of that all hormones. I hope it will be clear to all of you. Now after it, we have to study synthesis of steroid hormones. We have peptide hormones versus steroid hormone synthesis. How there is synthesis of peptide or protein hormones, and how steroid hormones are synthesized. Okay. So let us study. So steroid hormones can be transformed to active steroid in the target cell. Sorry. So we already discussed it before also, that this is a property of a steroid hormone, that sometimes what happens, instead of be an active form, they will remain inactive and they become active only when they reach the target cell. So this is the testosterone. So steroids can be transferred to active steroid in the target cell. So they will become active only when they will reach the target cell. Like this is testosterone. In one cell, it enters the cell because they are little soluble. So testosterone gets changes into dihydrotestosterone. So this testosterone, it gets changes into dihydrotestosterone. Now this dihydrotestosterone will bind with the receptor. This dihydrotestosterone will bind with the receptor. It enters into the nucleus and then it will become active. Getting my point? Similarly here, this testosterone gets changes into estradiol when it reaches inside the cell. Then it binds with the receptor and it will lead to change in the activity of whole cells, or you can say from inactive form they will become active. So the functioning of this single hormone in different cell is different. Like here, testosterone gets changes into dihydrotestosterone. So the functioning of this cell will be totally different from this cell. However, the precursor is the same. The main hormone or the main steroid is the same, testosterone, but the functioning will get changed. From here, in this cell, testosterone gets changes into dihydrotestosterone, and in case of this cell, testosterone gets changes into estradiol. So their functioning will be different from that cell. So this is cell A, this is cell B. So the functioning gets changed. That is why the steroid hormones generally they will remain in inactive form, and they will become only active when they reach the target cell. Are you able to get it? So the steroid cells, steroid hormones generally remain inactive and they become active only when they reach the target cell, because their functioning will be different according to the target cell. After it, steroid hormones are non-polar, that means they carry no net charge and thus can diffuse through the lipid membrane. We know it that they will pass from the membrane. They leave cells shortly after the synthesis. So when they are formed, when they are synthesized, they will act on the target cell as soon as possible. Getting my point? So they will act on the target cell. So as they are non-polar. But the polar substances are water soluble and the non-polar substances like steroids, they are lipid soluble. Now, functioning of steroid hormones. That what is the need of these steroid hormones? We have to discuss all these things, okay? So, steroid hormones play a role in carbohydrate regulation. Hello? Hello? मैं क्लास ले रही हूँ अभी तो 2 घंटे की मेरी कंटिन्यू है मैं आपको 1 बजे तक कॉल करूँगी हाँ थोड़ा जरूरी फिर क्या हुआ नहीं मैं लेक्चर दे दूँगा 20 से 15 मिनट की ब्रेक दे दूँगी ऐसा करती हूँ ठीक है So steroid hormones they play an important role in carbohydrate regulation because they are either glycocorticoids, mineralocorticoids or gonal steroids. They play an important role in carbohydrate regulation, mineral balance, reproductive function. We have already discussed these in detail. Glycocorticoids, mineralocorticoids and gonal steroids. Then steroid hormones also play a role in inflammatory responses, stress response, bone metabolism, cardiovascular fitness, behavior. So that is why this is very important thing that sometimes what will happen, we are become very aggressive within 1 or 2 minutes. Okay, sometimes what happens, we are very happy and immediately we feel very sad. This happen in our daily life. Sometimes we feel so bad if he or she, your friend or someone is saying you something, we feel so bad because there is a change in our behavior and even in our mood. So all these things are under the control of these steroid hormones. So of course they play an important role in carbohydrate regulation, mineral metabolism and even in our reproductive functioning of our body. But instead of it, these are the another things, but instead of it, steroid hormones play a role in inflammatory response, stress response, bone metabolism, behavior, cognition and mood. So our mood, our behavior, the fluctuation in our mood, the fluctuation in our behavior, all these are under the control of these steroid hormones. Now, how do the synthesis of steroids differ from the peptide hormone? Let me check it whether you are getting it. So is it clear to all of you up to now? Is it clear to you all? Please, up to now, is it clear to all of you? Are you feeling comfortable or are you feeling bored? Please let me know. I think hormones are very interesting topic. If you will discuss, if you will able to understand it in detail, then they are quite helpful to all of you. Are you feeling bored or are you feeling happy? Please let me know. Anamika, Anamika, why are you taking this address? You have to send me the abstract on my mail ID. I am sharing them. I already shared my email ID so many times, but I will share it again. No worry. Don't take stress or something of these review papers or all these things. Please be relaxed. Okay. Even if you want to take 1 or 2 days more, you can. No issue. So you have to send me the abstract along with the topic on my email ID, Anamika. So don't worry regarding it. So are you able to understand it or are you feeling bored? Everything is fine. Have you done steroid hormones before also? Have you done hormones before also, like adrenal hormones? We have pituitary hormones. We have hypothalamic hormones, thyroid hormones. Have you done it before also? Any one of you, please let me know. Because we are discussing very basic things. I am just letting you know from the very basic. Okay, so you have done it before also. Please reply, all of you, have you done it before also? Okay, okay, so you have done it, okay, good, good. But we are discussing very basic things which are there in the hormones because our main focus is on disturbances or abnormalities because of these hormones. Okay, so you have done these things in your endocrinology part before. Okay, okay. So you must have endocrinology in your syllabus or some topics will definitely be there. So let me complete it now. So now, how does the synthesis of steroids? They are different from the peptide hormones. How the synthesis of steroids? They are different from the peptide hormones. Now what happened here? Like while peptide hormones are encoded by specific genes, steroid hormones are synthesized from the enzymatic modifications of cholesterol. So in the case of steroid hormone, there is only the modification in case of cholesterol. So there are enzymatic modification of the ring structure of the cholesterol. However, in case of peptide hormones, they are regulated by the specific group of genes. So they are regulated by the specific group of genes. So thus there is no gene which encodes aldosterone. For example, as a result, there are far fewer different types of steroid hormones than peptide hormones. So peptide hormones, they are encoded by a specific group of genes, but in case of steroid hormones, there is only the modification in the cholesterol. So steroid structures are the same from species to species. So structure of steroid hormones will remain the same. The regulation of steroidogenesis involves control of enzymes which modify cholesterol into the steroid hormone of interest. So there are only a group of enzymes, there are only few enzymes which modify the cholesterol into the steroid hormones. So when there is little bit modification in the ring structure of the cholesterol, it will lead to change in the steroid hormone. Either it will be testosterone, it will be estrogen, it will be progestogen. So there is only the modification in the cholesterol by few enzymes. So only few enzymes are there which will modify the cholesterol. So the role of cholesterol in steroid synthesis. The first enzymatic step in the production of any steroid hormone begins with the enzymatic modification of the cholesterol. So here is only the modification of the cholesterol with the help of few enzymes. So the sources of cholesterol was steroid synthesis. So cholesterol can be made within the cell from acetyl coenzyme A. This is a multi-step process. It involves many enzymatic reactions. So first of all, what we need to synthesize cholesterol. So from acetyl coenzyme A, we will able to get the cholesterol, because if we want to synthesize steroid hormones, we must have to synthesize the cholesterol before. So cholesterol can be synthesized from acetyl coenzyme A. This is a multi-step process, involves many enzymatic reactions. So this involves from acetyl coenzyme A, we will able to get cholesterol from many steps. So a key rate limiting enzyme is HMG-CoA reductase. So this is the enzyme. This reductase enzyme plays a very important role. This reductase enzyme plays a very important role in regulating or in synthesizing the cholesterol. Now there is a negative feedback regulation of this reductase also. So there is a negative feedback regulation is also by the cholesterol itself. So when the level of cholesterol is high, it will negatively or it will feedback the enzyme to stop your more production. Getting my point? So for steroid synthesis, we need the cholesterol. Cholesterol can be made from the enzyme, can be made from acetyl coenzyme A with the help of an enzyme reductase. So reductase is the one which will helpful for us to synthesize cholesterol, and there is a negative feedback also to stop the further production of cholesterol when the number is quite high. Now, what happened after it? So, first step in the steroid synthesis is the formation of, or in the synthesis of cholesterol with the help of enzyme. Now, when we get the cholesterol, what happened now? Cholesterol is also taken up by the cell in the form of LDL. So yes, this is the thing. LDL is low density lipoprotein, and we have HDL also, which is high density lipoprotein. So this is your homework. You have to let me know what is LDL or what is HDL. What is the difference between low density lipoproteins, and what is the difference between low density lipoprotein and high density lipoprotein? That means the difference between LDL and HDL. You have to let me know regarding it. So, cholesterol is taken up by the cell in the form of low density lipoprotein. This LDL is a complex composed of cholesterol, phospholipids, triglycerides and proteins. Okay, proteins and phospholipids makes LDL soluble in blood. So, this LDL is soluble in blood. So, it will pass inside. So, LDL is taken into the cell by LDL receptor and broken down into cholesterol, and then free cholesterol will be released out. So, LDL is quite important for the steroid synthesis. Let me discuss it again. That cholesterol which we have just synthesized here, we just let you know how to make the cholesterol from different enzymes. So cholesterol is taken up by the cell in the form of LDL. So first it is taken up by the cell in the form of LDL. LDL which is soluble in the blood, so it will pass into the cell easily, and then it gets changes into the cholesterol, and then the free cholesterol will be released out from the cell. Getting my point? So the amount of free cholesterol in the cell is maintained relatively constant. So this is the whole thing inside the cell. LDL will be taken inside the cell in the form of LDL. Then it gets changes into esterified cholesterol. this esterified cholesterol, right, like here, it will changes into free cholesterol. Okay, so this esterified cholesterol will changes into free cholesterol, which will help in the synthesis of all steroids, which will be helpful in the synthesis of all steroids like testosterone, progesterone, all steroids. This will be quite helpful. Getting my point? So this LDL is taken into the cells via LDL receptors. So these LDL receptors allow the LDL to pass inside and broken down into esterified cholesterol, and then free cholesterol will be further used for steroid synthesis. So LDL is taken inside, it gets changes into esterified cholesterol, which further changes into free cholesterol, and then we are able to get the steroid which we want to synthesize. So this is basically how the synthesis of cholesterol is, the synthesis of steroids basically is different from the peptide hormones. Now, cellular localization of the cholesterol metabolisms for the steroid production. So, the first enzymatic step is in steroid synthesis is the conversion of cholesterol into pregnenolone. The enzyme that catalyzes this reaction is located in the inner mitochondria membrane. So this is how we are able to get another different types of steroid hormones from the cholesterol. But you have to keep in the thing which in mind is you have to keep this thing in mind that cholesterol gets changes into steroid hormones, but with the help of few enzymatic reactions. Getting my point? Cholesterol is able to change into steroid hormones, either testosterone, either glycocorticoid, mineralocorticoid, estrogens, progestogens, but with the help of few enzymatic reactions. So, these are the major pathways in the steroid synthesis. These are the major pathways, like from cholesterol we are able to get pregnenolone, we are able to get progesterone, we are able to get corticosterone, aldosterone. So but there are some enzymatic reactions which takes place. Getting my point? So there are some enzymatic reactions, there are some enzymes like we have desmolase, we have dehydrogenase, we have hydroxylase, we have beta hydroxylase, aldosterone synthase, aromatase. So all these enzymes are there for the production of specific steroid hormone. So there are the enzymatic reactions which takes place during the steroid biosynthesis. Are you getting my point? So there are some enzymatic reactions which takes place during the biosynthesis. Now, this is again the same thing, steroid hormone synthesis, that all steroid hormones are derived from cholesterol, we know it. A series of enzymatic steps in the mitochondria and in the endoplasmic reticulum convert cholesterol into the steroid hormone. We discussed it in detail. After it, the rate limiting step in the process is the transport of cholesterol from the cytoplasm into the mitochondria. This step is carried out by steroidogenic acute regulatory protein. So this is a protein which is steroidogenic acute regulatory protein, which is responsible for the transport of cholesterol from the cytoplasm into the mitochondria, because the synthesis of steroids, it takes place inside the mitochondria or the inner membrane of the mitochondria and endoplasmic reticulum. So free cholesterol has to be transported into the mitochondria with the help of this protein, which is steroidogenic acute regulatory protein. Okay. The cholesterol precursor comes from the cholesterol synthesized within the cell from acetate from cholesterol ester stored in the intracellular lipid droplets or from uptake of cholesterol containing low density lipoproteins. So lipoproteins taken up from the plasma are most important when steroidogenic cells are chronically stimulated. So these regulatory proteins, they are quite important for the synthesis of steroids. So if you will be able to see, this is the cholesterol. This is a free cholesterol which moves inside the cell. This is the protein, steroidogenic acute regulatory protein. So this cholesterol will be received by this protein. Then it will come inside the mitochondria, and inside the mitochondria, this cholesterol will either change into pregnenolone, progestogen or testosterone according to the, you can say, according to the requirement which is there in the body. So the cholesterol will be received by this protein. It will changes into in the mitochondria either into pregnenolone, into progestogen or testosterone according to the need which is there in the body. So these are the some functions of hormones, that means the function of progesterone, function of glycocorticoid, function of mineralocorticoids. As we already know that mineralocorticoids or aldosterone, they maintains blood volume and blood pressure by increasing the sodium reabsorption. So they maintain the salt and water intake in the body. So aldosterone is responsible for the salt and water intake which is there in the body. Glycocorticoids, they promote glyconeogenesis, favor breakdown of fat and protein. Progestogen, what progestogen do? It helps in the preparation of the uterus lining during the when the mother is going the birth. At that time progesterone is quite helpful. So this is the function of these all hormones. We have progestogen, glycocorticoid, mineralocorticoids, then we have androgens, estrogens and vitamin D. So they have development of male secondary sex characters, development of female secondary sex character in case of estrogen. Okay, so these are the important points which are there in case of steroid hormones. So from cholesterol we will get progestogen, from progestogen we will get aldosterone, and from progesterone we will get cortisol. So there are but some enzymatic reactions which are takes place with the help of enzymes like hydroxylase. So are you getting it up to now? Please let me know, are you getting it? So please let me know, is it okay? Are you getting it or not? Okay, so let me complete it then. Let me complete it, then I will ask you the questions regarding the review paper. Let me complete this first. So from cholesterol we will able to get pregnenolone. From pregnenolone we will able to get progestogen, or we can say progesterone. Similarly, from it we will able to get testosterone, estradiol. So all these things are there by the enzymatic reactions which takes place. So pathways for the synthesis of testosterone and estrogens and testosterone. So these all are the pathways for the synthesis of steroid hormones basically. Now, this is the biosynthesis of vitamin D3 and its metabolism. Like from the diet, we are able to get the vitamin D3, and in the liver, it gets changes into 25-OH-D3. In the kidney, it gets changes into active hormone, and then it gets changes into 7-dehydrocholesterol. And then from the skin, when we will get the UV light, when we get the UV light, or from the sunlight, we will able to get the vitamin D3. So this is the biosynthesis of vitamin D3 and its metabolism. Basically it is responsible for the reabsorption, or you can say reabsorption from the body. So this is 1,2,5 dihydroxy vitamin D3, which we have discussed also. This is also derived from cholesterol and it is lipid soluble. It is not really a vitamin because it is synthesized. It is synthesized not by body but by outside also. So it acts as a true hormone. So instead of considering it as a vitamin, we will be able to consider it as a hormone. 1,2,5 dihydroxy vitamin D3. Now this is again the same thing. From cholesterol we are getting progesterone. From progesterone we are getting corticosterone and aldosterone. Similarly, from this cholesterol we are able to get the cortisol. But all these things are happen with the presence of enzymatic reactions. So we have enzymes 2,1-hydroxylase, aromatase. So all these enzymes are quite helpful in the synthesis of steroid hormones. Now this is the transport of the cholesterol. That cholesterol is lipid soluble and mostly located within the external mitochondria membrane. So the conversion of cholesterol to steroids occur in the internal mitochondria membrane. Okay. So this is a regulatory protein which is steroidogenic acute regulatory protein which transports cholesterol into the mitochondria, moving it from the outer membrane to the inner membrane. So this protein is quite important. Please keep it in mind. Steroidogenic acute regulatory protein. We have already discussed these. Yes. This is the organization of the adrenal gland. I am not going to discuss this in detail, but only I will cover a little bit part of it, that there is an adrenal cortex and adrenal medulla. So if we are discussing about adrenal cortex, that we are saying melanocorticoid, glycocorticoid and androgens, they are from the adrenal cortex. There are different layers of adrenal cortex. We have zona glomerulosa, zona fasciculata, zona reticularis. From all these different layers, we are able to get these hormones, which is melanocorticoid, glycocorticoid and androgens. This is again the enzymatic reaction. The enzyme 17 alpha hydroxylase lyase, 3 beta hydroxysteroid dehydrogenase, 21 hydroxylase, 11 dehydroxylase, 18 hydroxylase oxidase. So these are the enzymes which are involved in steroidogenesis. Now what is steroidogenesis basically? When we are able to get the steroid hormone, it is known as steroidogenesis. So this is basically these are the enzymes which are important in this steroidogenesis, or for the function or for the synthesis of steroid hormone. This is the organization of the testis, function of laying cells, which is responsible for luteinizing hormone with the steroid production. We will not. This is the pathway of testosterone production in the testes. So the production of androgens from the cholesterol is identical to that in the adrenal, except that it continues from the androstenedione to testosterone. So from androstenedione, we are able to get testosterone with the help of an enzyme which is 17 beta hydroxysteroid oxidoreductase. 17 beta hydroxysteroid oxidoreductase. This is a metabolism again, that how it can be converted further. This is again the this LDH will come inside with the help of receptors. We get changes into cholesterol, and cholesterol will lead to androgen. Now this androgen from the theca cell is moved towards granulosa cell, and this androgen then changes into estradiol. So this is two cell theory of estrogen production in the ovary. So from one cell, that is from the theca cells, we will get the androgens, and these androgens when enter into the granulosa cell, these androgens will get changes into estradiol in the presence of enzyme which is known as aromatase. Getting my point? So in the presence of enzyme aromatase, we are able to change androgen into estradiol. So this is two cell theory of estrogen production in the ovary. This is a regulation of ovary hormones. From cholesterol we are get Pregnenolone, and from it we are getting the progestogen. There are some hormones also which are playing important role, like follicle stimulating hormone and luteinizing hormone, which will enhance the expression. This is the cortical effects, the body response to stress. What will happen when we are taking excess of stress, when we are worrying about everything? What will happen in our body? So, permissive effect on glycagon, memory, learning and mood. There is a swing in the mood, glyconeogenesis, muscle breakdown, lipolysis, immune depression, circadian rhythms. All these things are effective when you are under the stress. When you are taking the stress, what will happen? It will also disturb your sleep also. So, the circadian rhythms are also disturbed. So you will be able to see this is the body response to stress. So if you are able to see how the concentration of cholesterol will increase or decrease, how the concentration will decrease. So at the midnight the cortisol level is very low. During the midnight you can say that the cortisol level is very low, and at that time, that means in the midnight or 11 to 12 p.m. you can say, we are more prone to stress or any type of depression or something in the night, because the level of cortisol is very low, and the level of cortisol increase in the morning. Okay, the level of cortisol is increased in the morning up to noon. Then after noon again, after 3 p.m., after 2 p.m. or 3 p.m. again, we are feeling the low. So we are feeling high in the morning. According to this graph, we are feeling high in the morning, and we are feeling low in the noon, and we are feeling very low in the midnight. Getting my point? So this is the graph. Now this is the control of cortisol secretion. There are some feedback loop also. So definitely there are some feedback system when we want to stop the excessive production of any type of hormone or something. There are some feedback also. So these are the diseases which are related to cortisol. Use of immunosuppressants. We are having Cushing syndrome, Addison disease. All these are related with the cortisol only. These are again some important features of steroids that they are made from the cholesterol are lipophilic and enter the target cell. You know it. They are immediately released from the cell after the synthesis. Yes. So they are not stored in vesicles. Interact with cytoplasmic or nuclear receptors. So they can pass from the membrane and they can interact with the nucleus also. Activate DNA for protein synthesis. They are slower acting and have longer life than the peptide hormones. So they act very slowly, but they keep persistent. So they have longer half-life than the peptide hormones. The example is estrogens, cholesterol and testosterone. This is again the steroid hormones, how they are working inside the body. Steroid hormones molecular action, how from the bloodstream they enter into the nucleus, into the cell membrane, and then it enters into the nucleus, and they will do the changes here in the cell. So you have to keep these points in mind. So first of all, what will happen, that they will diffuse from the blood and they will enter into the cell, and inside the cell they will bind with the receptors which will either be present on the cytoplasm or they will be present inside the nucleus. So these steroids, these yellow ones, these steroids will bind with the receptors. So these yellow ones are steroid hormones. They just enter here from the bloodstream. So these receptor hormone complex binds to a DNA and activates or suppress one or more genes. Now this hormone will bind with the DNA and it will either activate or react the activity of other genes. So other genes create mRNA that move back to the cytoplasm. So we will able to get the mRNA from the DNA which will move back to the cytoplasm. Translation produce new proteins for cell processes. So we will able to get the new proteins. So some steroid hormones also binds to membrane receptors that use second messenger system to create rapid cellular response. So they bind with the membrane receptors to create rapid cellular response. So binding with the receptor is quite needed here for rapid responses. So they will bind with the cell surface, they will bind with the cellular receptors for rapid response. So this is the main functioning of the steroid hormones. I hope all of you will get it. I will share the PPT, then you will do it by your own self also. These are the functions of steroid hormones: carbohydrate regulation, mineral balance, reproductive function, inflammatory response, stress response, bone metabolism, cardiovascular fitness, activates DNA for protein synthesis. All these are the functions of the steroid hormones. So please let me know, is it clear to you? Are you getting it properly or are you feeling bored? Please let me know. Is it okay? Are you able to get it or not? I will share the PPT with all of you so that you will able to understand the steroid hormones in detail. Okay, I will share it with all of you. So did you get it? Everything is fine? No, please, yes, please let me know. Give me the status of your review paper. Before starting the new topic, please let me know the Status of your review paper. So yes, we have done protein hormones, we have done steroid hormones. Now we have to do these disturbances in the thyroid and metabolic and hormonal disturbances. We have to discuss these things. But let me know whether you have, what is the status of your paper. First let me know about the status of your paper. All of you please let me know. Have you decided the topic? Have you write the abstract or not? Please, all of you, please let me know whether you have selected any topic or not. Please let me know. Please share the topic of your review paper. All of you, please. The status of your review paper. All of you. Okay, conservation of coral reef ecosystem. Okay, okay. Need of the hour. Okay, okay. Very good, Anjali. Conservation of coral reef ecosystem. Yes, Ayushi, please share the topic if possible right here. Please share the topic only. The name of your topic only. Afternoon is epidemiology of hepatitis B virus infection and prevention. Yeah, good, very good, Afri. Please keep it up. The topic is very nice. No, Anjali, there is not any limit of referencing you have to use. At least you have to go through 9 to 10 review paper. That is the only thing. Okay, you see oral delivery of anti-cancer drugs. Okay. Archana's topic is clinical biochemistry of pregnancy. Oh, very good, Archana. Koshali also sent me the mail also. Inborn errors of amino acid metabolism. No. Your title is good. No worries. Inborn errors of amino acid metabolism. So yes, the topics you selected are very good, and I will suggest you to read at least 9 to 10 review papers if possible, so that you will have, you know, good content for your own review paper. Getting my point? So I want that you have a good content for your own review paper. For that you have to read many papers, at least 9 to 10 review papers. Even you can read research paper also, no worries, but you have to read the paper and you have to take the important points from that particular paper. You have to take the points that what author wants to tell us. From his or her paper, what the author wants to tell us, you have to take those results and you have to add those nutshells in your own review paper. Like we can say Smith et al. 2016 had these results. So you have to take their work and have to put the nutshell of your work in your own paper. So I request you all, it's my suggestion from my side, that if you want to take more time, please take more time, but your content should be good. Otherwise, you will send me the paper of 5 to 6 pages. When we will send it further for publication, they will not accept it. That is my point. So our paper will get rejected. So please, it's my suggestion to all of you to read your papers, to read the paper of previous work, to write your own review paper. Otherwise, the paper will get rejected. After so many of efforts, after so many of work, the paper will get rejected if we will not make the content good. Yeah, monoclonal antibodies, clinical diagnosis in brief and its application. So you have to not to write in brief, write only monoclonal antibodies, clinical diagnosis and its application. But please remove in brief. You have to write monoclonal antibodies, clinical diagnosis and its application, or you can say clinical, write it like that, clinical diagnosis of monoclonal antibodies along with its application. So you have to write clinical diagnosis of monoclonal antibody along with its application. Or you can say clinical diagnosis of monoclonal antibodies and its application. Okay. Suradippa is saying botanical description and pharmacological study of, what is the common name of this? Cabra montana diversificata. What is the common name of this, Suradipa? A review. Description, botanical description and pharmacological study. Please add the common name in the bracket also. After the scientific name, please add the common name so that all will be able to read your abstract or your review paper with interest. Okay? Please go on. Okay. So Namita, if you are listening, please make those changes in the title if possible. So you have to write the common name after the scientific name. Have you selected the review paper? Have you downloaded the review papers or not? Have you downloaded the review papers related to your topic or not? Yes, so your topic is very good, Suradipa. Topic is good, but I want that your title should be attractive also. All of you, your title should be attractive. Okay, so you have downloaded it, okay. Otherwise, take your time and download it today so that when you will be free, you will be able to read those papers. So please, it's my suggestion to all of you to download the paper as soon as possible and try to read at least one paper daily. Try to read one or two review papers even in the night or in the when you will be free. So try to read one or two review papers daily so that you will be more concentrated that what you want to write. Are you getting me or you are not getting me? So I will suggest you to read at least one or two review papers so that you will have a knowledge the things which you want to write in your own review paper. So even in the night before going to sleep, at least try to read something related to your own topic. I'm not want to you to study the whole day. No, just take 10 to 15 minutes only or 15 to 20 minutes only. Take those 15 to 20 minutes only for your topic. Are you getting me? Please let me know. Just give 15 to 20 minutes to your topic. That's all. 15 to 20 minutes are more than enough. Yeah, even before going to sleep, when you will be free, just take 15 to 20 minutes only. So are you able to get me or not? Please reply. Please reply if you are able to. Yes, Pooja, if you want to add the images, then you have to add the reference also. That from where you have taken those images. Okay, so if you are taking the images from someone, then you have to add their references also. Okay, so we have done protein hormone, steroid hormone. Now I will start enzymes. Okay, I will start now enzymes. Enzymes are very easy. So I try to start a little bit today and we will try to complete these things in the remaining days. So we will start with the enzymes. However, day 3 and day 6 is still pending. But I know when I have to complete it. So I will start enzymes now. Okay. So let me share the PPT of enzymes with all of you. And I will share the PPT of steroid hormones with all of you. Okay, let me close it. We have to just take some introduction related to enzymes, then we will wrap it up. Okay, so I'm just going to start little bit of enzymes, what are basically the enzymes and their nomenclature, their history and their classification. So these are enzymes. You all know about it that enzymes are, yeah. So enzymes are biological catalysts that speed up the rate of biochemical reaction. So enzymes are basically what? They are the catalysts which speed up the rate of biochemical reaction. Enzymes are three-dimensional globular proteins. Some RNA species also act as enzymes and they are known as riboenzymes. Now this is your homework. You have to let me know what is the function of ribozymes, or you can say why RNA are working as enzymes. So you have to let me know the function of ribozymes. So this is your second homework. First you have to tell me about LDL HDL, difference between LDL HDL, and now you have to tell me about ribozymes. So enzymes are biological catalysts that speed up the rate of the reaction. The enzymes are three-dimensional globular proteins. Some special RNA species also act as enzymes and they are called riboenzymes. Enzymes are biological catalysts synthesized by the living cells that accelerate biochemical reactions. The orderly course of metabolic processes is only possible because each cell is equipped with its own genetically determined set of enzymes. It is only this that allows coordinated sequences of reactions involved in many regulatory mechanisms. Almost all enzymes are proteins. So basically enzymes are proteins, but there are some exceptions where RNA acts as the ribozyme. So you have to tell me what are basically ribozymes. Now this is the history of enzymes when we have started the enzymes. So enzyme was coined in 1878 by Reticulum coining. So it was coined in 1878. Biological catalyst that has previously been called ferments. So enzymes are previously known as ferments. Later they are known as catalysts, and after it they are known as enzymes. So the enzyme is basically comes from the word in and yeast. In means enzyme means yeast. So it literally means is in yeast, because most recognizable reaction properly known as alcohol fermentation by zymase enzyme in yeast. Why these are known as enzymes? Because they are first recognized in alcohol fermentation by zymase enzyme in the yeast. So this is its history. It was came into existence by 1878. Early it was known as ferments, later known as catalysts, and yes, they are the enzymes which are found in the yeast. So this is the history of these enzymes, how they are able to know about the enzymes. Like there was the scientist in 1830 who prepared malt extract from germinating barley seeds. So he was able to prepare malt from the germinating barley seeds. Similarly, we have another scientist who prepared the enzyme diastase, also known as amylase. Then we have another scientist who prepared a substance from germinating wheat like diastase. So they are able to get pepsin, trypsin. Slowly and slowly we are able to know about these all enzymes. Then in 1926 we have another scientist, James B. Sumner, at Cornell University isolated and purified an enzyme, the urease. So he was able to isolate urease, thus confirming the proteinaceous nature of that particular enzyme. So basically enzymes are protein in nature, but some are exceptions also. So enzymes are key to life. No enzyme, no life. So no enzymes, no life. But how and why? We have to discuss it. So what is an enzyme? Yeah. So enzymes are basically the globular proteins which function as biological catalysts, speeds up the reaction. Okay, so enzymes are basically biological catalysts which speeds up the reaction by lowering the activation energy. So enzyme speeds up the reaction by lowering the activation energy without being affected by the reaction it catalyzes. So it will not affect the reaction, rather it will enhance the reaction. It will speed up the reaction by lowering the activation energy. So these are the enzymes which are proteinaceous in nature. We have globular protein, we have riboenzyme, we have some enzymes which are present in the inactive form also. Like zymogen and proenzymes, they will remain in the inactive form and they become active only when they are needed. So now what is basically the active site? So enzymes are composed of long chain of amino acids that have folded into a very specific three-dimensional shape which contains an active site. So we have a active site. An active site is a region on the surface of an enzyme binds to which substances will bind and catalyze the reaction. So this is the enzyme. Enzyme consists of an active site, and on this active site the substrate will bind. Okay, so we have the active site on which the substrate will bind. So enzymes are highly specific for the type of reaction they catalyze and for their substrate also. Enzymes with higher specificity and accuracy are involved in copying and expression of a genome. These enzymes have proofreading functions, example DNA polymerase. So, some enzymes require non-protein molecules for their activity. So, enzymes are conjugated proteins known as holoenzymes. So, basically what is holoenzyme? It consists of a apoenzyme plus a cofactor also. So, these cofactors increase the activity of the enzyme. So, enzymes without a non-protein part is inactive. So, apoenzyme is basically inactive. It becomes active only when the cofactor is attached with it. So when the cofactor is attached with the apoenzyme, it becomes active. Now this is the mechanism of enzyme action. The enzymatic reactions take place by binding of the substrate with the active site of the enzyme molecule by several weeks. So enzyme will bind with the substrate and it will lead to the formation of the product. So enzyme just enhance the rate of reaction. So formation of enzyme complex in the first step in the enzyme catalyst reaction. It forms ES complex which later gets changes into enzymes plus protein. So enzymes will be free and we are getting the product. So E plus P means enzyme plus product. So from the substrate we will get the product. Now structure of enzyme. The active site of an enzyme is the region that binds substrates, cofactors and prosthetic groups and contains residues that help to hold the substrate. So active sites generally occupy less than 5% total surface of the enzyme. So active site has a specific shape due to the tertiary structure of the protein. So active site which we have, let me show you, active site is the site on which the substrate will bind. It is further divided into two types, binding site as well as catalytic site. So on the binding site the substrate will bind and on the catalytic side there will be a reaction. So if you are able to see here, this is the inactive protein or you can say apoenzyme. When cofactor will bind with it, it will get changes into active protein. And active protein has the binding site for the substrates. Getting my point? So active protein has a binding site for the substrates. So what are these cofactors which enhance the or which activates the proteins? What are these cofactors? They may be inorganic cofactors or organic cofactors. So inorganic cofactors include like zinc, iron. These are basically the inorganic molecules required for the proper activity of the enzymes. So examples are carbonic anhydrase require zinc, or you can say hexokinase enzyme. It is the apoenzyme, or you can say inactive enzyme. It needs the cofactor magnesium. Similarly, we have some organic factors also which is needed for enhancing the reaction or for activating the enzyme. Now we have another term which is there. We have prosthetic group and we have coenzyme. Now what is the prosthetic group basically? So prosthetic group is a organic compound. I think it's not written properly. So a coenzyme is loosely tightly bound organic compound, or yeah, there is some mixing of these lines. Let me show here. So an enzyme with its cofactor removed is known as apoenzyme. So apoenzyme is basically what is the inactive enzyme. When the factors, when the inorganic or organic factors will bind with it, it will become the holoenzyme. So if you are able to see here, the holoenzyme or holoprotein generally consists of organic molecules which are attached with it. So these are the, if you are able to see here, these are the protein chains. And we have some active sites also. So what is basically substrate? Substrate is the one which binds with the enzyme and it will form enzyme substrate complex. So enzymes are synthesized by ribosomes which are attached to the rough endoplasmic reticulum. Information for the synthesis of enzyme is carried by DNA. Amino acids are bounded together to form specific enzymes according to the DNA codes. Now we have intracellular and extracellular enzymes. So some enzymes are there which will remain inside the cell, like oxidoreductase. They are found in the cytoplasm, nucleus, mitochondria and chloroplast. But some extracellular enzymes are there which are synthesized in the cell but are secreted from the cell to work externally. So we have some enzymes which are there in the cell but they will be released out. Like we have some digestive enzymes which are not used by the cells in the pancreas but are transported into the duodenum. So they will be transported out. So we have some digestive enzymes which are produced by the pancreas and they will be transported into the duodenum. So we have intracellular and extracellular enzymes. So these are the characters of enzymes. I think you all must know it. That enzymes speed up the reaction. So enzymes speed up the reaction by lowering the activation energy. Their presence does not affect the nature or the end product. So they only enhance the reaction. They will not change the functioning of the product. Okay, getting my point? So they will only enhance the reaction, rather they will not change the working. They are highly specific in the action, but that is each enzyme can catalyze one kind of substrate. Small amount of enzyme can accelerate the chemical reaction. Enzymes are sensitive to change in pH, temperature and substrate concentration. So enzymes are quite sensitive to the pH, temperature. Turnover is defined as the number of substrate molecule transformed per minute by one enzyme molecule. So these are the examples, like for lactose we need lactase enzyme, for maltose we need maltase enzyme, for protein we need protease enzyme. So this is a lock and key mechanism which is there in between the enzyme and substrate complex. So this is known as ES complex and we have lock and key mechanism. So this is induced fit theory, that how substrate will bind with the active site of the enzyme. So this is the induced fit theory. I will discuss it in detail. This is a nomenclature which is there of enzymes. Let me check it just once. Okay. So I just want to say all of you that we will start from the induced fit theory which is there in case of enzymes. So what happens in induced fit, basically we have the enzymes and enzymes have a specific active site. So substrate will bind with that particularly active site and then they will form the enzyme substrate complex which is there. So tomorrow are you able to get it or not? Are you able to get it or not? Please let me know. So this is induced fit theory which is there. So in the induced fit theory, we have the enzyme which will bind with a specific protein, in the induced fit theory, we have an enzyme which will bind with the specific substrate. But these substrates have to bind with the active site of the enzyme. So, and one more thing which I want to show you, this thing, that enzymes work only so what enzymes do, they just lower the activation energy. If you are watching here, The reaction is this: H2O2, H2O2, we are able to get O2 and water. So when the enzyme is not here, the activation energy is quite high. The activation energy is quite high, but when we are using the enzyme, this blue one, the activation energy gets low down. So this is the thing. We are able to get the reaction fast by lowering the activation energy. So there is the low down of the activation energy during the whole reaction. Are you able to get it or not? So when the enzyme is absent, the activation energy is quite high. When we are using the enzyme, the reaction is fast and even the activation energy is also less. So are you able to get it? I think that's all for today. Otherwise, you will feel bored also. We will discuss tomorrow also enzymes. We will start from the enzymes tomorrow. But up to now, is it clear to all of you? Please let me know. Up to now, is it clear to all of you? Otherwise, if we will do much more, you will feel, you know, little bit stressed. So we will not doing it that much. We have just started the enzymes. What are basically the enzymes? What are the properties of the enzymes? But other things we will discuss tomorrow. Is it clear to all of you? So tomorrow, this is your third homework which you have to tell. You have to tell me about induced fit theory in case of enzymes. So you have three homework, I think now. Please, any one of you write the homework so that all of you will be able to do it. Okay, please write the homework which you all have to do. The difference between STL and LDL, number one. What is number two then? Ribozymes. What is the difference between, what are basically the ribozymes? And number three, you have to let me know about the induced fit theory in case of enzymes. So is it clear to all of you? All of you please reply. I am just going to stop it right here now because otherwise it will become too much. So is it clear to all of you? Please let me know. You have to tell me about induced fit model in case of enzymes. Induced fit model. Please let me know about the homework, all of you. So you have to do all these things and you have to let it to know me. Okay, so what's your homework? Any one of you please write it here. I am waiting, just write it here. So we have just started the enzymes today. Tomorrow we have just discussed the history of enzymes today. Tomorrow we will do the nomenclature and classification along with other things in the enzymes. Okay, so we will do the nomenclature and classification tomorrow. Up to now, is it clear to all of you? Please let me know. Is it clear to all of you? Please let me know. Kindly reply. So that's all from my side. We will meet tomorrow. Till then, take care of yourself. Afternoon, you are very right. You have to tell me what are basically the riboenzymes, why they are functioning as enzymes, Also, they are not the proteins, but why they are functioning as enzymes. What is the difference with what is basically LDL, LDP, which is low density protein, and SDP, and what is the difference between these? And then another thing is you have to let me know about induced fit model in case of enzymes. So this is your homework. We will meet tomorrow. Till then, take care of yourself. Take care of your own self and of your own family also. And yes, best wishes from my side. Take care. So enjoy your day. So you have whole day after this noon. So please, thanks for being here along with me for such a long time. Thanks for your patience. Thank you all. Thank you all. Thank you so much. So if you have anything to say, you can ask or you can let me know. Yeah, yeah. Please take care of yourself. All of you, please take care. Take care of yourself, all of you. If you have any query or question, you can ask me. No worries. If you have any doubt or something, you can ask me. So very thank you for your patience and we will meet tomorrow. Please complete the homework in the meantime. Yes, we will meet tomorrow.